of Medicine in Boston. She is a specialist of endocrinology and developmental biology, and also cancer, oncology in general. As you can see here, she has also an affiliation with the Centre Cavaillès at the École Normale Supérieure in Paris. Anna has worked a lot with Carlos Sonnenschein, who will be mentioned in a minute. Carlos happens to be Anna's husband and main collaborator, so we'll hear more about this in a minute. Anna was Blaise Pascal Chair of Biology at the École Normale Supérieure some years ago, and she has interacted with many biologists, also historians of science and philosophers of science, which was also something interesting for us. In 1987, with Carlos, they showed the mimetic action of estrogen by plastic products, what is now called endocrine disruptors. As you might know, because I think my email went to all of you many times, tomorrow Anna will give another conference on endocrine disruptors in French at the Athénée Municipal. Today, Anna will tell us more about one other aspect of her work, which sometimes puts together endocrine disruptors and cancer, but more generally, this is focused on cancer and on the idea that we don't have a good theory for understanding cancer. And one of the things that Anna did with Carlos was to try to provide a theory for cancer based on different ideas, like the question of cell proliferation, or what is the default status of a cell. And as you will see in a minute, one of the main aims of what Anna does, which I think is extremely interesting, is to put together different branches of biology. So it can be about cancer, embryology, endocrinology, all that put together in a way which I think makes very good sense and is very interesting. So one of the reasons why we wanted to hear Anna is both to hear something about cancer and also to show that if you want to think about cancer, it's extremely important to think very broadly about this pathology with different domains of biology that are very useful to think about that pathology. Anna, thank you very much for having accepted our invitation and for making it with the trains in a period which is a little bit complicated in terms of transports.
Okay, well, thank you very much, Thomas. But I also would say that I support the Les Revistes de Vive la Rêve. It was a little anguishing, okay, because I thought that maybe we couldn't make it. But at the same time, I mean, I wasn't upset because I think that that is the thing. I mean, I love France, as you know. I spend lots of times in France. And one of the things that I love about France is the sense of preserving democracy, because things tend to entropy. And what is happening right now is this preservation of democracy. So, I mean, some people may think otherwise, but for someone that is living in America, in a moment that is so prone to fascism, is very refreshing. Although I was also here in 1995. Okay, but I didn't come for that. I just happened to be here. Anyway, so I would like, first of all, to thank Thomas for inviting me, because I love to talk about this theoretical experience that I had, and I'll show you a little bit of my parcours, because I changed the title from Experimental Carcinogenesis to A Theory of Organisms and back, because we started with a theory of cancer, and then in time, we went to a theory of organisms, and now we talk about cancer in the context of such a theory. Since biology is a historical science, I think that it's also important to talk a little bit about the parcours, because ideas don't happen because one follows a logic all the time, but there is this part about life that takes you somewhere. And so here I would start then with experimental work that initiated all this. In 1971, Carlos Sonnenschein, by the way, is not my husband.
Oh, sorry.
Okay, but yeah. Anyway, so Carlos established the first estrogen target cell lines, and these cell lines were called responsive to estrogen because injected into animals, they only grew if you give estrogens to the animals. So, according to the standard theory, the target cells proliferate when they are stimulated by hormones and remain quiescent in the absence of estrogen. So he took those cells, injected into the animal, and this is what he got. No estrogens, no proliferation, estrogens proliferation. However, when put in culture, they proliferated equally well with or without estrogens under our in culture conditions. So, you see there is this paradox. And so these experiments gave us a paradox, and that is the most beautiful gift that you can have, because that will tell you that something is wrong and will show a way out, probably. Or at least that is how Niels Bohr thought, and in this case I agree. So we have to start to think at that point, how is that estrogens regulate the proliferation of their target cells? And here is our conditions. You have these cells that are estrogen target and in the animal require absolutely estrogens to proliferate, and you put them in the presence of absence of estrogen in a plasma serum-free condition, and they divide. They don't care, they divide, and estrogens don't do anything regarding proliferation, but they induce progesterone receptor, for example. Fine. So after many years of working on that, we found out what was going on. Well, if you add to your serum-based medium plasma or serum, they are inhibited, as we are showing here. No proliferation. And when you add estrogen, there is proliferation. So there was something in serum that inhibited the proliferation of these cells. And so we purified it, and it turned out to be serum albumin, of all things. And then we synthesized it and found that domain 1 and domain 1+2 have that activity and not domain 3. Well, and we stopped there. But we got to see that the organism has a way to stop these cells. And then we had to think, why is that? And so we started talking, well, what is the default state? And we knew, because it's an axiom in microbiology, that prokaryotes and unicellular eukaryotes have as default state or as constitutive state proliferation. Provided that you give them nutrients, they proliferate. And when you remove nutrients, either they die, or they sporulate, or they do something like that, or just, but if there is nutrients, they proliferate. They don't need any sort of so-called signal. And there is where we started to have very good rapport with our friend Kupiek, who were talking about, that those signals don't exist as so-called signals. In metaphyta, in plants, you have the very same story, and you can do the experiment yourself. I mean, what you do is take a leaf, you crush it like this, cells are liberated, and you put some light and carbon source and nitrogen source, and bingo, you get a plantlet. So, why is that we, working with animals, think that cells are quiescent? That is what the dogma was. Well, as I said, all these unicellulars and plants, default state, as an axiomatic is proliferation and motility. And we thought, well, our experiment showed that metazoa maybe is the same, it's proliferation and motility. And we started looking at the literature to see where this idea of quiescence started. And it started by some practitioners of tissue culture, because actually in the 19th century, at the end of the 19th century, it was the general understanding that the cells constitutively proliferated in animals, even in animals. And then there is, this is an aside, but Lenny Moss wrote about the change in mentality that occurred at the beginning of the 20th century that has to do with all these changes in his books, What Genes Can Do. And I think that is very, very interesting because people seem to have forgotten all this Kantian way of thinking and adopted a totally different outlook. And they became Darwinians in the bad sense. The bad sense is that what happened is that agency, I mean the normative agency of organisms was removed from organisms and stuck on natural selection. So now organisms became like a little thing, like a rock, that you have to do stuff to them. I mean, instead that they would do stuff themselves. Okay, so at that point we decided, well, if data are not theory free and there is no theory of organisms, what needs to be done? And we were thinking a lot at that time. Dan Dennett, he's our colleague at Tufts, was always trying to tell biologists that data don't speak by itself, but you know, they are very, they think that they can observe things directly, etc. And so at that point we decided that what needed to be done is that we have to start to think theoretical. And so we took a sabbatical, and that was in 95, and then in 99 we published this book, The Society of Cells, that was translated into French, in 2006 was published, and in 2019 it was published in Spanish, and now it's being published in Italian, and we are thinking of a second edition in English and French as well, with the changes that we have introduced both in the French and the Spanish edition. So by then, that is by 2003, we had a sabbatical, I had a sabbatical at the École Normale Supérieure, and we published the Milieu Nigène. So at that time, there was also a lot of work that we did there in the frontier between theory and philosophy. And then when I got the Pascal, I organized the organism group. My host was Giuseppe Lomo, a mathematician, and so there are some philosophers there that you may know, that Paul Antonio Michele and Matteo Mosio, and then biologists like Carlos Arnaud Pocheville and I, and Maël Montevilli, that is a little bit of each one, or a lot of each one, because he's a mathematician, a physicist, a biologist, and also has a degree in philosophy, master's. So, and he works in at the confluence of all these disciplines. And so we produced an issue towards the theory of organisms where we talk, we explain these principles, and I'm going to talk about that briefly to give, or not so briefly, to give context to our view of cancer. So I should start, why do we need theories? And probably, I don't know who this audience is. I suppose that there are biologists and philosophers. And probably biologists that are in contact with philosophers have an idea about what do we need, why do we need theories, but I have to put that in case there are people that don't. Because in one of the study sections I participated, people, I mean the study sections in America judge grants, okay, grant applications. And so they say, bring data. Okay, so we only trust God. So bring data. What does data mean without theory? And so I started educating some of these people, but they don't want to be educated. So anyway, we need theories for many reasons, but probably one of the fundamental ones is that it provides organizing principles and construct objectivity by framing observations and experiments, and we don't have direct access to objects, so we have to do that through a theory. And also theory determines which are the proper observables. I mean, the Newtonian physics don't mention that the atom is an observable. It doesn't exist in that theory. So this penchant in biology for reductionism, that everything has to be explained molecularly, is something that predates Poincaré, for example. So it's a very rigid idea that comes from, the 18th and 19th century physics, and naive at that. Now, the true choice of observables is also a major theoretical commitment. And I like to give you the example that when you decide to investigate the relationship between pressure, volume, and temperature, like in thermodynamics, you do that independently of the microscopic structure of matter. And that is so important. We have to think of that all the time, because in biology, nobody can understand that there are things that are only explained well at the mesoscopic level, for example. So, but what we did is to learn about theories by looking at theories in physics. But physics is different in many respects from biology, or vice versa. And Giuseppe Longo says that biology becomes physics when we are dead. So I think it's quite interesting. So a theory that is biological is different from a theory in physics, because we have problems that we cannot apply the ideas that we have in physics. For example, invariants. There are not invariants and symmetry conservation in biology. Biology is science of change, both in the long scale of evolution and in the short scale of organogenesis, or say directly life cycle. Okay? And also, we have to be bold, like physicists, in picking up a limit case, because that is how modern physics started, and any part of physics. You have to pick up your postulates or your principles and build from there. And in our case, we want to say that in a way our default state is like inertia in physics, in classical physics. At the same time, besides the awareness that in biology, you don't have a predetermined phase space, because in evolution you don't have that. I like Stephen Jay Gould saying that if we play the tape of life, rewind it and play it again, we will get to different species. Okay? So, and also the awareness that biological objects are normative agents. And so we have a very strong French tradition on that. Bichat wrote a lot about it in his very short life. And Canguilhem did very excellent work on that as well. And I would say that we can go back to Nietzsche to understand this aspect that is so fundamental in biology. So the default state in mechanics, and we call it default state because that is how we call ours, but it's basically the principle of inertia. If no causes modify the properties of the physical object, the object conserves its properties. So, inertia and the default state is what happens when nothing is done to the intended object or system. And also, we have to think that inertial movement is a limit state. Galileo made it intelligible. And so, what he says is that if you accept inertia, then you can focus on the analysis of the forces that constrain inertia. Okay? And so that was our goal in establishing a principle of default state. So, now we should concentrate in the biological default state. So for a theory of organisms, we have proposed three principles: the default state, that is the one that we first worked on, Carlos and I, a principle of variation, and a principle of organization. And I'll start with the default state, that is the unconstrained state of cells in the presence of nutrients is proliferation. with variation and motility. Carlos and I were talking about proliferation and motility, but talking a lot with Giuseppe Longo and Mael, we decided to include the idea that variation is also generated there, because you can see how two cells are not identical. So although variation occurs at different levels of organization, even at this very elementary one, that is the division of one cell into two, variation is created. And this is rooted in the cell theory and in the notion that cells are agents that generate their own changing rules. Now, this default state also is present in evolutionary theory. So not only the cell theory provides a bridge between organisms, that is ontogenesis, and evolution, but also it's basic to the thought of Darwin when he was talking about descent with modification. So he made explicit the concept that living entities reproduce following a geometrical progression. In his words, there is no exception to the rule that every organic being naturally increases at so high a rate that if not destroyed, the Earth would soon be covered by a progeny of a single pair. So we think that it makes sense in the short scale of an organism and in the long scale of evolution. And I would say that our proposal has straightforward consequences on what requires an explanation in the sense of a theoretical cause. The default state does not require such a cause, like the inertia principle is. You put it forward is a principle. To the contrary, a departure from the default state. That is, if a cell is quiescent, if variation is restrained, if there is lack of motility, that requires a causal explanation. And the default state, proliferation with variation and motility, expresses, again, normative agency and modifies the causal structure with respect to the theories of the inert. And there I go back to the conversation that Thomas and I had before the seminar, that for Kupiec, this variation sort of is just the presence of the random. And for us is this proto-agency of cells, and that would do stuff. And if this agency is normative, you can get wrong. Okay, so something is going on there that could look random, and it could have elements of randomness as well, but there is also this agency that counts on this lack of Laplacian determinism in what is biological. So I will speak a lot less about the principle of variation, that is a source of novelty, but Longo and Monteville wrote extensively over that, saying that it's fundamental to the differences between physical objects and biological objects. And they created the concept of extended criticality, because they compare this ability of the organism to change with the phase transition. And so they say we posit that the dynamics of biological organisms are not just processes, but permanent, critical transitions, and thus symmetry changes. Within the limits of a relative structural stability, variability is at the core of these transitions. And of course, it's the very source of novelty that we see through evolution, and also in individuals that, like animals that are born without the front legs, they can, quadrupeds that walk on their rear legs, for example, that is just showing this normative agency, and also this possibility of creating novelty, because the pelvis of those animals changes to become, in the muscles, to imitate what we find in bipeds. So the relentless change addressed by the principle of variation points to the major difference between the theories of the inert and those of the living. And so the consequences of the principle are historicity, so contextuality, variability of biological objects. And I'm not going to say much about that, but I'm just going to show an example because it has a lot of bearing on how we do biology and the fact that statistics is a mathematical discipline, for example, and we are applying statistics in physics. It's about errors of measurement, and here is about variation among individuals, which is totally different. It's plus the errors in measurement. So these are the mammary glands, the buds of the embryo at day 15. This is the mouse. And this, in the same animal, would generate in 3 days this structure, or this structure, or this structure. Okay, so there is a lot of variation, and in spite of which, and for those that are going to go to the endocrine disruptor talk tomorrow, we can put this together with the effects of bisphenol A. You see, my controls are like this. The bisphenol A also vary a lot, but I can distinguish between the two using classical statistics. So it's more difficult because this is what you see. That is why you need a bunch of animals, not just a few. And then the principle of organization, that is a source of stability. So it's there where you get this organism to have a way not to break apart each time that it sneezes or something. So we need not only that creation of novelty, but also a source of stability in our theory. So the notion of closure of constraints is the means to achieve and maintain stability, and this was traditionally applied to intracellular processes, but Mossio and Monteville explored this concept by taking in principle, this is Monteville is a mathematician, so if we put the constraints at a different time scale than the process is being constrained, we can model it mathematically. And this works very well. This concept opens a point of entry for the mathematization of biology, and we exploited this notion to model mammary gland morphogenesis, using as principles the default state and its constraints. Because one of the things that people don't realize is that when we do mathematical modeling in biology nowadays, what we are doing is we are transposing physical principles or physical equations that seem to represent in a way our biological model. And this is what Turing used to call imitation, because real modeling comes from principles. So the idea was, can we model from these principles? And I'm going to talk very, very succinctly about that a little later, because in any case it's published Monteville et al. And 3 years ago. So the principle of organization works by separating processes that are physical and chemical changes that occur in biological systems, but they also involve, well, all of them, alteration, consumption, and production of entities, like a chemical reaction. That is what has been studied the most, or morphogenesis, as Mael and us have done. And constraints are entities which, while acting upon these processes, can be said to remain unaffected by them, at least under certain conditions. So in our time scale, our constraints don't change, while the processes do. And they have this clever diagram, okay, where the arrows from, say, A3 to C2 indicate a process. And then these squiggles are the constraints. So we have a C3 constraint that is constraining this transformation. And in turn, this product here, so is a constraint for A2 to C4, etc. So you get this closure of constraints, and so one becomes dependent of the other, and you close this circle. And in a way, this is a representation of what can't talk about this circularity in organisms. But that will bring us stability, of course. And we are now using this principle to model, for example, the control of glycemia instead of using a feedback with a lot of interesting ramifications. So now that we have the three principles, we can talk a little bit about organogenesis. And during organogenesis, the default state results in tissue growth by generation of new cells and the production of extracellular matrix. This process is constrained by physical determination involving flows and forces, and shape is a result of this default state, constraints like gravitation and the constraints created by living organisms like muscular tension. But if you look at early embryogenesis, you will see a lot of movement and motility of these cells anyway. And from the very beginning, you start seeing how physical forces are created at the supracellular level. The ability of an organism to generate new constraints at each new iteration generates diversity, even that iterations are not identical. So that is what you see. I mean, you observe that when there is a branching process, the angle of the branches is not constant, for example. And that depends on the fiber arrangement around those ducts, for example. So we were working, now this is experimental, on what are the determinants of shape. And here we show you, this is, if we have a matrix for a three-dimensional model of proliferation, we use a matrix that is collagen, that is totally five-regular, that allows cells to use this to move along them and to create forces. Or you have a collagen matrigel. Matrigel is a extracellular matrix, like basement membrane. So you see that this matrigel is covering those fibers. It's the same amount of fibers that you have here. And so we operated with this two types of matrix and observed the following, that if you have a lot of matrigel, what you have is rounded acini, that is the structure predominant in the mammary gland of an animal that is lactating, nursing. Or you have, if you have very little of matrigel and a lot of collagen, I mean the collagen is constant, you have branching ducts. And we try to, this is a very long story, we determined that there were many variables there, that is rigidity, fiber organization, etc. But we decided to model it mathematically and see what it gives. And so in this model we have a biological component that are the cells and a physical component that is the matrix. So the cells constrain each other and they exert forces that organize the collagen matrix, and if there is basement membrane proteins, that is sort of blocked in part for that for those roller proteins, by those roller proteins. And then as collagen organizes, constrains the proliferation and motility of cells. And this is, I'm going to go very, very fast through that. So what we observe, I mean modeling this, the lumen, that is when the duct becomes, ceases to be a cord, to have, to be like a tube, occurs when the structure is a little bigger. But the first thing we see is that as soon as these cells start proliferating, those are the orientation of the fibers, they change. And so these cells organize the fibers in a way that goes along the duct. If we look at what happens in real life, in light microscopy, what is interesting is that by 3 hours you know what is going to happen. Because, sorry. I took the wrong one. So you see the cells are moving and start getting elongated and picking up other cells. So that means this is the same matrix in one place, and then I'm not doing the acini, but if you have at the beginning the cells can grab fibers, they would start forming a duct. If in the immediacy these fibers are covered by a globular protein, they will form an acidos. And so what we learned from this modeling is that with the default state and closure of constraints, we could generate a bistable situation of two outcomes, round versus elongated structures. And in order to get them, we had to create an inhibitory layer to constrain cell proliferation and motility in order to prevent the open-ended growth of epithelial structures. And this goes with theory because of what I told you before of the default state for estrogen-sensitive cells that is constrained by plasma-bore inhibitors, but also is constrained by cell-cell interactions. The closure of constraints is operative at the tissue level of organization, so it's not only good for metabolic processes. And now we will change scope and go finally to neoplasm. For that, I want to show you a microscope and a normal tissue and a cancer coming from this mammary tissue. And as you see, what the pathologist, that is the only person that diagnosed the cancer, would say is that there is a big tissue organization change that not only affects the epithelial cells, but also the matrix that is there. So it's not only a change in the epithelial cells. Now, the classical theory that we have since 1914, the somatic mutation theory, says that if you get the carcinogen will go to the nucleus of an epithelial cell, produce mutations there, and those mutated cells now cannot control proliferation anymore, and they will proliferate. So the mutation is to make them to proliferate, you see, because they are this default state is quiescence, according to these people. So, the heritability of the neoplastic phenotype is due to somatic mutations in the DNA of a single cell. Carcinogenesis is the problem of the control of cell proliferation. The default state is quiescence, and that is the theory that I stopped there because we published enough and others showing that this theory is full of holes. So let's look instead to our proposal. And to go to our proposal, we have to start with organogenesis very briefly, because we say that this is the problem of organogenesis, or at least of the morphogenetic field and the interactions that happen there. This is in the embryo, and was done many years ago in 1987. Sakakura took embryos and dissected with enzymes the epithelium and stroma of the mammary gland and the salivary gland. So, if we put the epithelium of the mammary gland in the salivary gland and the epithelium of the salivary gland in the stroma of the mammary gland, what you get is that the structure, the epithelial structure, obeys the effect of the stroma or mesenchyme. In other words, here we have epithelial structures that were remodeled to imitate mammary gland, and they came from the salivary gland, and here we have the epithelium of the mammary gland that now is imitating the salivary gland. And it happens in many organs. I'm not going to give you the list, but it happens all the time. Morphogenesis depends on these interactions in the field. To the point that in the kidney, for example, and this is what I would like to stress the most, you have two organs, two origins, the nephrogenic mesenchyme and the Wolfram duct. Now the mesenchyme, if you label the mesenchyme, you will see that there are 10 cell types that come from it. So you need this very close contact in order to have everything. So it's a lot of movement and interaction that will generate the nephron, as you see here, but also 10 cell types, different cell types will be formed, like the mesangial cells, the inner cells of the Bowman capsule, etc., the cells of the macula densa, etc. And all this cytodifferentiation is not primary. It depends on tissue differentiation. So it's the opposite of what people would think. And if you do it in culture, by putting the cells in culture, you would never get out of the mesogenic mesenchyme all these 10 cell types. Okay? So this is why cells are part of the tissue, but they are not determining what is going on. It's the tissue that is determining it. That of course contains cells, but without these interactions, the inner part of the cell won't do what it's doing. It's not, in this case, a spontaneous, it's not like a cell living on its own. And so the tissue organization field theory says that cancer is a problem of tissue organization akin to organogenesis. And so if the NOxa, instead of being the carcinogen is not just hitting the epithelial cells, it's hitting the tissue. And in the case, for things that you are going to see later, it's affecting mostly the mesenchyme. And when this happens, these bidirectional interactions are affected. As a consequence of these alterations, the cells in the epithelium start to be less constrained, so they will proliferate and organize differently and move. So you see, the default state is proliferation and movement. Now you don't have to explain metastasis and proliferation, like in a tumor, as things that are acquired because some growth factor is inducing them. Is something that happens because constraints are not acting as they were acting before. And so the problem happens at the tissue level of organization. So the target is, if you will, just to screw up these interactions, the double-headed arrows. That is what you're doing. Cancer is a problem of tissue organization comparable to organogenesis. As if we disrupted the interaction of these two tissues in the kidney, we will have something similar. The default state of cells is proliferation with variation and motility, and this is how we see it. And now a little clarification, because this seminar was, by chance, put after your publication in eLife. So, I'm going to that, just 1 second. So, I enjoyed reading the paper, but there is something that we have never said. That is this. It's so ingrained. Everyone has to explain that these mutations are causal. And we say, this is an epiphenomenon. We never attributed causality to them. Okay? So this is just a clarification. Mutations are an epiphenomenon irrelevant to carcinogenesis, and oncogenes are expressed in normal tissues. And by the way, what was interesting is that in the critique to the TOF, it says that, why do we need mutations, says the paper. Well, we don't. Anyway, so, but we can discuss that later. So, as I said, cancer is on tissue organization. And here we have a control where these epithelial cells are forming a duct. The duct has to transport milk to the outside, right, to the exterior. That is the duct function. And the ductal carcinoma has all these complications, like cells are growing inside the duct, blocking it here and there. So what do you have here is more complexity, because here you have, topologically, you have a single lumen, and here you have many lumina, okay? But here, this simplicity is functional, and here this complexity is not. So you have a dissociation between complexity that is totally anatomical, and organization that is both anatomical and functional. So, as I said, I was putting the arrow, or we were putting the arrow, mostly on the stroma, based in the sense that the interaction between the two tissues experimentally was affected because we exposed the stroma. And I'll show you how we did that. In the mammary gland, if you use a carcinogen called an MU that has a half-life of 20 minutes, and it's an alkylating agent, people have written, you give an injection, intraperitoneal like this, and the cancer appears in the mammary gland. And so they say it's mutating the DNA of the epithelial cells of the mammary gland. And we said, oh, really? How do you know? So what we did was, okay, let's take the mammary gland, separate the epithelium from the stroma, and then expose individually the epithelium and the stroma, knowing that the half-life is 20 hours. And so we changed the medium many times and waited 5 days, and then did the recombination. Okay? And the recombination was done in a set of 4 recombinations. Vehicle with vehicle, NMU with NMU, and then crossing. Okay, and here you see the results. Okay, if you recombine cells, let's see, well, this is in the animal, intact animal, you inject NMU and you have negative control, no tumors, positive control, 100% tumors. And then what you do is you do the recombinant. Like vehicle, you don't get anything. If you expose the stroma to vehicle and the epithelial cells to NMU, you get nothing. If you expose both to NMU, you get tumors. And if you expose the stroma to NMU and the epithelium to vehicle, you get tumors. So this is compatible with the TOF, but it's not compatible with the somatic mutation theory. Okay? And the corollary of this idea that the mutations are an epiphenomenon is because you can normalize cancers. You dissociate the cells of the cancer and you inject them in normal tissues. There are 50 papers already that show that. And they reacquire function and form part, like say, of the liver or mammary gland, etc. In our case, the experiment that we did was before those appeared, but so what we did is quite complicated. What we did is took animals, and when they were 10 days old, we removed the epithelium from the mammary gland, so that now we could inject epithelial cells and see what happens. And then what we took is different tumors from several animals that had tumors. So it's a lot of logistics, because the same tumor has to be, you separate the epithelial cells and you inject at 24 days of age, 52, 80, 150 of virgin animals, and 2 animals that were already twice parous. Okay? The idea is that this is prepuberal, this is immediately postpuberal, young adult. This is kind of middle-aged adult, when the mammary gland is quite quiescent. And this is 150-day-old, is an older animal. And this is an animal that has had litters and nursed them already. So the point is that here in normal life, if there were epithelial cells, they would be proliferating like crazy, and in here they would not. And but we are counting on the stroma directing this orchestra, okay? So what happened? Well, what happened is, here you have the primary tumor. When we took this and injected at 27 days of age, you got a secondary tumor that is more aggressive. I mean, the end was 10 or so I'm just showing 1. If you injected it into a 52-day-old, you got an anaplastic tumor, even worse than this. And here, instead of showing the section, I'm showing the whole mount of the mammary gland. So the 83-day-old animal produced with cancer cells a normal mammary gland. And so did the animals that were twice parous. So the stroma could get those cells and organize them in normal structures. And we are not alone. There is a guy called Gil Smith that has done that in many instances. And So, and it happens also in the liver, it happens with melanoma injected into embryos when the neural crest cells are migrating. Well, there are at least 50 papers that show that. So, a group of normal cells in the wrong morphogenetic field may generate a process akin to carcinogenesis in an animal. This is what we did on the recombination with NMU-treated stroma. Carcinogenesis in an animal may be prevented or reversed when tumor cells are put in contact with normal cells. So which is the conclusion now? We have proposed principles for a theory of organisms that provide an appropriate framework for the study of development and cancer. This theoretical framework provides means to perform mathematical modeling from principles rather than using applications from engineering and physics. This theoretical approach explains carcinogenesis as a problem of tissue organization, akin to morphogenesis gone away. And from the Tufts perspective, cancer is a process that can be healed by reversal to normalcy, by reorganization of constraints. And I should say that, well, I'll leave it for a discussion. So now what I want to do is to thank the organism group, Giuseppe Longo, Paul Antoine Miquel, Maël Monteville, Matteo Mosio, Nicole Perret, Arnaud Pocheville, and Carlos Sonnenschein, and also the NIH, the Yvonne Foundation, the ENS Foundation, the government of France, and everyone else that supported my research through these last 20-some years. And last but not least, I thank you for your attention.
Thank you very much, Anna. So we have time for questions. Maya first.
Thanks, Anna, for the talk. So how can you reconcile, the aspect of cell death with your theory and mutations that prevent cell death resulting in tumors independent of this interaction with the stroma?
Well, I would say that I have to see the context in which that happens, because there is a very interesting mathematical model of that was done with apoptosis in cancer. And actually, if you have prevention of apoptosis, you have less cancer formation than with apoptosis. This is a paper by, wait a second, I will remember his name in a moment. It's mathematical modeling. He's now in, I will remember and tell you later. These ideas that Weinberg had and were put as these hallmarks are not such. That is what I would say. And that you have to look exactly how these experiments were done, because it's not clear at all that there is evidence that shows that stopping apoptosis will have a worse cancer. Actually, there is no. I mean, there are cancers that have very little apoptosis and are aggressive. So how do you know?
Like from the chromosomal translocation of MYC and BCL2 and the
Well,
hormones,
there are many cells that have the same translocation and are not cancer, like megakaryocytes, for example. And there are people that carry those cells with the translocation that never develop cancer. So I don't know. I mean, at least I'm agnostic, but I have seen that. You see, the problem with the somatic mutation theory is that what Richard Feynman used to say, a vague theory. If you have a vague theory, you can add elements every day, because the theory says that cancer happens in a single cell when it stops having control for cell proliferation, and that is done by oncogenes. First it was 1, then 2, then 4, now it's a myriad, and the opposite is anti-oncogene. So it starts adding elements, and nothing is removed. So a good theory, Poincaré said, if you have a good theory is a theory that you can do something about. I mean, if your data is against your theory, now you have to think, where is the problem? So it's a global problem, because your experiment is telling you the whole theory is wrong. So now you have to look at the different postulates. But if you have a theories for that, not for adjusting more stuff to it, and then I'm right in any case. So you cannot, it's a very funny thing if you have a theory that can never be rejected because it has so many components and some of them are one the opposite of the other. Yeah.
If I build an organism according to the three principles you have explained, I'm concerned that it would probably never die except by accident. Is there any way of showing, or is it an additional principle that is needed to, or required to explain that it should die, or?
Well, I think that you have the organisms also, I mean, they are creating, they are discharging entropy in creating organization. So entropy doesn't disappear, it goes somewhere. And I think that that somewhere depends, like the matrix, like call it the elastic fibers get affected by that. I mean, they are not replaced at the same rate, and they cannot be degraded totally. The glucosaminoglycans of the articular surfaces, by the time we are training, they are just not as resilient as before. And if you are a runner, they get spoiled before. And so there is all this problem with the idea that tissues repair faster when you are young. So to me, that means that we don't have a very good way to canalize this entropy. And that in some systems, like the neurons, the older you are, the more lipotoxins you have in your brain. So just because of that, I don't think I have to look for a principle to kill entities that are alive. That is inescapable.
I have a question about constraints. So
Yes.
you say that cells are usually proliferative and mobile, and in some cases, many cases, they are constrained, I guess, by the context. And in cancer, especially metastasis, it's less constrained.
Mm-hmm.
There's something I really don't understand. It seems that in the first phase of tumorigenesis, this is super constrained. I mean, it's often taken as a good example of something which is super constrained. A tumor is very constraining. So it seems to me that what you say explains more something about metastasis, maybe. So we suppress these constraints, and then it's going to be very mobile. But in the first phase, I don't really understand why it's less. There are, less constraints in the case of a tumor than in a normal tissue.
Well, I think that is less constrained because if you see, I mean, I don't want to do circular arguments, and the problem is that Boveri was right. Nobody has seen cancer in tumors. What he said is nobody has seen the birth of tumors, okay? Because it's not like the orderly process of embryogenesis, that if you have sea urchins, like he did, put the sperm, and then you take your clock and now your time, and you know where to harvest them. Here you don't. But people have done interesting things. Like, for example, you take an MU and you give it to your rats, and then you look at, and it's a lot of work. I haven't done it, but what you do is you take the mammary glands and you start doing whole mounds, because the whole mounds you can see the whole thing. So you have to do young animals, but that is another story. That is those animals that can develop cancer fast. And so what you see is in the whole mound, that is the whole tissue clarified. You have your ducts there, the mixing, and you will see that this is the invasive part, that is the terminal valve, and you have proliferation here and cell death here, because here is where the lumen is going to be produced. Now, these are targets, apparently, of what is going on, but also some ductuli that appear here. So, at about 3 weeks, 2 and 3 weeks, you start seeing these little structures forming rounded things. So it's like the cells are growing towards the center, and then they are going in all directions. And you see a lot of proliferation, and not a lot of, here you see that there is still cell death. There is this duct, the lumen being formed. But they become bigger and with more proliferation. In all cases, when it's growing, it's secreting proteases. And actually, it's a relationship. In order to secrete proteases, you need the other cells and stromal cells. So what is unconstrained to me is that if you look a little further, it seems that these things have done like turning over themselves. You have this, and then it's like they are growing and growing. And as they are growing, if you look at the lumen, instead of being like this, it starts being like blocking here. I mean, we have done confocal microscopy to reconstruct them, and they are blocked in many areas. So there is a lot, to me, that is unconstrained growth, because in normal cases, you don't have these cells doing this stuff. So, in our experiment, when we did the modeling, there was a need to add, in order to get orderly ducts and acini, we had to add an inhibitory layer. And that represents, we think, we have to add mathematically something that inhibits there, because otherwise the round structures will become much bigger and they don't become that large. And in fact, if you look at how they evolve, they stop at a given time. Now the rounded structures proliferate in a more or less homogeneous way. If you count the cells that are dividing. But in a duct, you have division in these two. It's very orderly. So all this is inhibited. You don't see any proliferation in here. And so I think that those cells are more constrained because they have neighbors everywhere. And here you have the fibers that are pulling, and that seems to be helping in this possibility of migrating along them. I don't know, but I'm giving you explanations that are not so-called proven, but that go along with the organization of both the fibers and the epithelium. So to me, the unconstrained part is that you have this excessive growth. But if you take that, as we have done and as Smith has done, and put it in normal conditions, and one of his conditions, because he doesn't do that only, he has done that with funny things like seminomas, human testicular cancer. That doesn't have anything to do there, but in order to constrain that, if you put it in the stroma, it doesn't work. In our case, the stroma is good to constrain the epithelial cells of the mammary gland. What he had to do is put 5-fold more mammary epithelial cells, and for each one of these seminoma cells. And if you do that, and even better if you put 10x, then what you get is normal ducts that have the Y chromosome here and there, and they make milk. But you need double constraint. And he hypothesizes that these cells create a niche. That is a mammary gland niche. And so now those seminoma cells have to behave like a normal epithelial cell. We do not know much, but there is evidence that something is happening that is constraining. Now, when you get the animal pregnant, now there is proliferation again. So that being constrained is that you cannot move in every direction and proliferate in any place in particular. But now you are doing what is done in normal life with the constraints proper to that part of the duct. So we have to find out what these constraints are. Some of them are just cell-cell interactions. They form junctions, for example. And one of the cases is in Drosophila. Animals that cannot find serrate junctions develop hyperplasias instead of normal, how is that called? These discs, I mean, that generate the different organs, like the disc that generates the wing. So you take that, and in the animal that is mutated because it can't make those, it proliferates and shape doesn't happen.
And Peter?
Just maybe a follow-up question on this. So I have the impression that so far, or maybe in just what you pointed out here on the whiteboard, we're talking about physical constraints, but then when you talk about cell-cell interactions, that may be both contact interactions, but also exchange of molecules.
Yes, yes.
So, and because you started out with estrogens, I suppose you have that in mind as well.
Yes.
So could you tell us a little bit more about how, so do you think of regulatory networks with feedback mechanisms as constraints?
Yes.
Okay, and so how can they interact with the physical constraints?
Well, I mean, in this particular case, I didn't look at both things together. What I looked at is physical constraints by changing the arrangement of fibers or by interfering using rigidity, like different matrices. So that I did in my system. What I know is that if I take another model that we developed that is sensitive to estrogens, progesterone, and it, by the way, is the only one that does this, progesterone, estrogens, and prolactin. It's a cell line that everyone uses into the culture situations, and we decided to put it in 3D. And it was derived from a human carcinoma. And the interesting thing is that when I put estrogens only, it does, we cannot erase this. Where can I move the screen up so I can, since I cannot erase, I can draw somewhere else. Thank you, I don't know where it was. Okay. If I have E2, they form structures. They are not perfect like with the other model, but they form structures like this, elongated. Now if you put estradiol, and because progesterone doesn't act alone, so you have estradiol plus progesterone. Now they branch. They form something like this. Okay. And if I put E2 MASP and prolactin, so they start doing this. Okay. So those are like acini, and that is, I mean, this gland, if you look at the pregnant mammary gland, there is acinous formation. If you look at what progesterone does, it's branching. Now this, the matrix is just collagen. So what we did was to look at the structure of the organization of those fibers. So it seems that with estrogens, the fibers organize in a way that is different with this 2 and with this 3. So like here you would see the fibers going. Whereas here it's like more orderly like this. And what is interesting is that if you look at the collagen matrix at polarized light, you don't see anything because those are like little fibers. You cannot see it at the optical microscope. So at 24 hours after seeing an empty gel, or if I have a gel that has those cells. And so it doesn't matter what I put, I start having specks like this. Those are the fibers. And with time, they start organizing. So if you have these structures, eventually if they become ducts, that will be elongating like this, like this. So what is happening there is that the hormones somehow confer to these cells different physical properties in their ability to move and organize the fibers. So it's a sort of transaction, but I don't know the elements because I'm, for the time being, interested in mesoscopic level more than anything else, because I want to see cell interactions. Then we'll dissect what they are, but everything is screaming biomechanics. And I am also sure, because of another guy at Tufts that is called Mike Levine, that works on electrical gradients. You can also modify electrical gradients, you can modify shape. But we haven't done both at once.
Thank you.
So, you're welcome.
Yeah, I have my second question was on the link between the notion of a default state that you use in your refutation of the somatic mutation theory and the refutation itself. Is it really necessary to use that notion to make a case at least about the limitations of the somatic mutation theory, if not, and maybe even the proper refutation of it? Because I'm not so sure that it's necessary. I'm not very convinced actually by the idea that the default state is really something substantial. I think it's more like you're explaining the psychology of the people that are defending the somatic mutation theory rather than their actual claims. I don't think it adds anything to the refutation. So I think I would make two different cases and I would use Occam's razor and get rid of the notion of default state. And have you tried that? I mean, because I think your case would be even stronger.
I don't think so, because I look at the history of that. And one of the early papers, many, I remember the guy that was at NIH, he was talking about, I don't remember which growth factor, but one of the claims was that these cells are quiescent and need a growth factor to proliferate. And that when they get to be a cancer, so-called, because you put an agent that induces cancer, what you get is expression of more receptors that somehow they are more, they bind better, whatever is there, but they become like, they say they don't need the growth factors anymore, okay? So if you are claiming that you need the growth factors, you have a default state in mind because you are saying that they are quiescent. And you see it over and over. Somewhere appears, and you're right, some people they don't talk about that, but it's adjacent to the problem, because if you are saying that what you need in normal life is growth factors, and then you become, how do they say? They use some word that is autonomous of stimulation, because it gets it inside, because they modify their cell cycle, which I think that cell cycle and control of proliferation are two things different, but anyway. So every time that I read these things, even if they don't say, hey, the default state is proliferation, it is implicit in it, and in some cases it's totally explicit. So that is why I use it, and because I think that it has evolved into a very vague theory, because if you go to the hallmarks, you put anything under the sun, and then there is no way you can contradict it, except when you take it to the point as Heiko did. Heiko, I can't remember his name, family name. Heiko used the one on inhibition of apoptosis, and he showed that using several models, all mathematical, that what you get is, if you inhibit apoptosis, is the tumor will grow slower. It will be slower. So I think that those things are, in many cases, just wishful thinking, because nobody saw a tumor in status nascendi, first of all, neither us, not them. But we could do these recombination things and show that you don't need to act on the epithelial cell, that is, this is a carcinoma, so that is the element that, according to them, is faulty. So I think that the default state is important just because of my observations, but not in metazoa, but also because it's an axiom for every other cell. And I don't know, although molecular biology of the cell has a paragraph that I could, if I can go to one of my slides, I put it in the notes in case I needed to quote it, but I can paraphrase it. And in my book, the book with Carlos, he has put it there, that says, oh, yeast and bacteria and all that have the inherent property of proliferating in the presence of nutrients, but metazoan cells don't. Why is that? Because they need to be quiescent, so the organism is not a blob, so now they need growth factors. Okay, show me why. You see how such a big thing that didn't happen in Metaphyta happened. You have, if this evolutionary change happened, where is the evidence? And I never found it. It was like by decree, they say it's like that. Okay. Plus that none of those growth factors is a growth factor. EGF, what it does is spread the cells. So in the periphery, because now your colony, instead of in this big, is bigger, in the periphery you have more cell proliferation. And why they found that? Because they wanted to sell it to treat burn people. And so obviously people would say, no, this produces cancer. This is 20 some years ago when people were saying that the default state was the cancer people. So they were saying, no, what it does is spread. If we don't let it spread that much, there is no proliferation, and it's no proliferation in the center. So that is one. But also every knockout of epidermal growth factor, receptor, etc., produces morphogenetic problems. But if you take the skin, for example, of these beasts and put it in a dish, they have the same mitotic index than the animals that have perfect receptor. Okay. So, I don't know what to say, but I cannot, if it's implicit, it's there. Because you can see it through the conclusions. That now they are autonomous to growth factors. Well, okay, that means that if you are normal, you are not autonomous to growth factors, you are quiescent. No. And many of them have said that. Like Fogelstein has said that. It's one that I remember that did.
Okay, Anna, you want to defend your paper?
No, the general question about the role of the stroma in your
Yes.
theory. I was wondering whether you always theorized about the stroma as a whole, or if you thought about the different types of cells or components.
Well, we didn't theorize about the role of each type because what we tried to do is see where you have to put the carcinogen to get the effect. But what we see now is the microarrays, okay? Because when we did this experiment, we didn't do the sequencing, it didn't exist. We did microarrays. You see a bunch of interesting things. Some of them may have to do with tissue repair, like molecules that have to do with macrophages and stuff like that. We counted macrophages. You see, this is very difficult because you have to do at 5 days, 10 days. So you see that there are different types. There is mucosal macrophages, and I don't know what are the others, but okay, 2 types that have 2 different markers. Maybe someone knows.
So they know.
Well, so some of the immunologists there said, okay, you can use this and that marker. That is many years ago. And so we see that there are different waves of migration of different components there, and I think that you have a lot of tissue repair because those things are quite, those are alkylating agents, so I think that it's not like a nice caress, right? And in these stromal cells. But everything that we saw had to do with this type of thing. So I think that, at least at the beginning, so we think that it has to do with tissue repair. And also animals, what is interesting is that there are animals that are resistant to NMU. And when they are resistant to NMU, what you see is that they, at 2 weeks, they form this pre-neoplastic carcinoma in situ, and then by 1 month they disappear. And so the animals don't get a tumor. But up to that point, when the thing has to happen, that from this carcinoma in situ, some of them will develop the tumor, they never do. But if you inject the animals, instead of injecting them at 20-some days of age, you inject them first at 2 days of age, and then at 20-some, you get like a 10% of animals get to cancer. Now what we did was to ask the question, is the stroma what is healing? So we did recombinants of stroma from one strain and epithelium of the other and put it under the kidney capsule. And that is just a pilot. We could never finish that because it's very expensive, so we got it up to 2 months. And then it's what forms is too big, even the normal tissue, so you start having problems. So nobody gives them for more than 2 months. But what we observed is that the ones that had the stroma of the resistant strains had normal structures, and the ones that had the stroma of the sensitive strains had abnormal structures, but they were not cancers because we couldn't wait until seeing them. If we get maybe 500,000 will do, but I didn't get it yet, so yeah, that one only costed, I think, 200,000, but anyway. Yeah, it's very expensive to do these things. I mean, it's terribly expensive. But you see that there is something in the stroma that has to do with this resistance, and probably something that has to do with the capacity of tissue repair.
Okay, I think it's time to stop and thank our speaker. Thank you very much, Alan. So remember, if you're interested, tomorrow, endocrine disruptors, 7 p.m.